The Taxonomic Impediment, part of the Systematics and Biodiversity course offered by Leiden University in 2020. I'm Jeremy Miller. In this lecture, we will explore some of the practical problems for applying and building biodiversity knowledge, the importance of archiving specimens in natural history collections like Naturalis, dark taxa and other signs of the vast underexploration of biodiversity, legal aspects of taxonomic publishing, and moving toward the semantic web of biodiversity. The focus of my work is on biodiversity, especially in tropical forests, with emphasis on diverse taxonomic groups. My particular area of expertise is spiders. So I want to start off telling you a little bit about how this kind of research is done, and about some of the asymmetries in global biodiversity that present challenges to developing knowledge about the world's most diverse communities. It begins with scientists going into the field to collect data. In this particular case, my colleagues and I traveled to a remote site in southern Guyana, in the northeastern part of South America. We arrived by small plane on an unimproved airstrip near a village of YY Amerindians. We loaded up dugout canoes and floated upriver to a plot of forest that would be our home for the next 2 weeks. We used a variety of sampling methods to collect all the spiders we could find. We found big spiders like this one, tiny spiders like this one. The average spider species is just under 3 millimeters in total body length. Some strange-looking spiders like this one. All in all, we spent 300 hours collecting 6,000 adult spiders from this 1-hectare plot. These comprise 350 species. We counted the number of individuals for each of these 350 species in our samples. What we found is that most spider species in this forest were very rare, and only a few species had very high abundances. Nearly a third of the spiders we found were represented in our samples by only a single individual. I'll call these singletons. This fits with an almost ubiquitous pattern that we find in tropical biodiversity. Rare is common and common is rare. What does it mean that there are so many rare species in our samples? These are sexual organisms that need to pair up in order to reproduce. So the obvious question is, where is the nearest neighbor for all of these singletons? Is it in the plot and we missed it? Is it outside the plot? Remember, these animals are on average 3 millimeters long. How far do they have to go in order to meet each other? Or perhaps there is some kind of a temporal edge effect. Maybe some of these species were rare at the time of sampling, but were common in the weeks before we sampled or after we sampled. And maybe some of these rare species are simply in the wrong place, far from other members of their species. Or perhaps the underlying species abundance distribution of this spider's community is so skewed that this pattern is what we should expect from a sample of this size. Or maybe we're just lousy collectors. To investigate that possibility, we looked at rare species in nearly 50 large tropical arthropod surveys. These studies focused on spiders and various insect groups. For all these studies, we plotted the proportion of singletons against sampling intensity, which is the mean number of specimens per species, a way to scale and compare inventories of different sizes. Plotting these on a log-log scale, we find that the more intensive an inventory is, the lower the proportion of singletons. But because this is a log-log scale, what this means is that as the proportion of singletons goes down, we have to make larger and larger collections to make the number of singletons go down further. And just to see where we stand for the Guyana spider inventory, here we are right on the trend line, indicating that the proportion of singletons is exactly what we should expect from an inventory of this size. Up to now, I've mostly been telling you about biodiversity information according to the taxonomic knowledge framework. This tends to be organized more or less phylogenetically. Observations and comparisons focused on closely related, physically similar organisms wherever in the world they might occur. This emphasized rigorous comparisons between closely related species to discover the best characteristics for distinguishing them. But often in biodiversity, we want to make comparisons between 2 or more biological communities. Relevant context for this might be widely scattered across taxonomic literature, and while some taxonomic groups may have good taxonomic resources available, There are often groups for which taxonomic literature is incomplete. When you think about taxonomic research, perhaps you think of describing new species. To be sure, this is one aspect of taxonomic research that tends to engage the public. But it is worth keeping in mind that, especially for very diverse groups about which knowledge is incomplete, knowing whether or not you are looking at a new species requires knowledge of all the other species that have been described. In many cases, this is not a trivial effort and can take significant time and effort in a laboratory. It is rarely something we know during field expeditions. So in practical terms, our ability to apply taxonomic knowledge can be uneven. Take, for example, an inventory of the Caribbean island of Montserrat conducted in the mid-2000s. This was a taxonomically broad effort, including work on relatively well-known groups like vertebrates and vascular plants, along with more diverse and unevenly known groups like beetles. A total of 718 beetle species were sampled from the island. Some were recognizable as belonging to species with valid names. Some were clearly undescribed. But a large portion of the collection fell into this ambiguous category, where it's not really clear whether they were described or not. The primary explanation for this is a combination of deficiencies in the literature, no taxonomic revisions, descriptions and diagnoses not up to modern standards, lack of illustrations, and the decreasing number of taxonomic specialists available. When we are unable to apply taxonomic knowledge to a collection, we are experiencing a phenomenon known as the taxonomic impediment. In this case, specimens from the inventory were archived in an institutional collection, so at least they can be available for future taxonomists producing revisions. But this may be years or decades away. For another example, let's look at all the Caribbean islands for which we have lists of spider species. These generally have 1 to 2/3 of the species identified with Linnaean names. The rest of the species can be called morphospecies, which tells us something about the total number of species known from each island, but complicates comparisons between islands. One of the key things we might like to know in a region like the Caribbean is which species may be restricted to one or a few islands, that is, island endemism. Assuming identification is correct, we can use Linnaean names to determine which species are shared among various islands. But with morphospecies, this is usually not possible. Invasive species, endemic species, species new to science, none of these come with labels telling you their status. The investigator has to be able to reconcile what they find in a place with other knowledge about those same species, including where else they have been found. So once again, the name, along with its synonyms, is key to accessing what is known about a species. In 2015, Naturalis sent a group of researchers and students to the Dutch Caribbean island of St. Eustatius. The aim was to produce an inventory of life on the island for a wide variety of taxonomic groups, covering a broad range of the habitats available on the island, from tropical forest on the rim of a volcano to an old yard in town. Here are the 11 quarter-hectare sample plots, each within a different vegetation type. A consistent sampling protocol was applied to all plots, designed not to discover every species present on the island, but to produce comparable samples across a broad range of taxonomic groups. Here are the first 10 datasets of this project, including plants, vertebrates, gastropods, and arthropods, plotted as the number of species against total sample abundance. Thousands of specimens and observation records contributed to this study. For all the taxonomic groups that we studied, we attempted to identify all records to species. But for those groups where the taxonomic impediment is most severe, this was not always possible. So we created libraries of images of all species for these taxonomic groups: ants, beetles, true bugs, spiders, and snails. This was supplemented by DNA barcode sequences that can help scientists determine whether species on different islands are the same or not. Some species are identified with taxonomic names, others are not. But we can use the image library to help us recognize when we next encounter a species we have seen on St. Eustatius. This can help us monitor changes in the biodiversity of St. Eustatius in the face of impacts from more frequent hurricanes and climate change. They can also help us better understand which species are shared with nearby islands and which may be unique to St. Eustatius. Knowing which species live where is key for setting conservation priorities. For example, in 2003, Madagascar announced that it would triple protected land. But to determine exactly which areas were going to be newly protected, the government asked for assistance from scientists. So a group of researchers took data on 6 taxonomic groups: ants, butterflies, frogs, geckos, lemurs, and plants. Most of these data were extracted from field guides and taxonomic monographs. The data on the ants, one of the groups frequently associated with the taxonomic impediment and not typically available for this kind of assessment, was extracted from the AntWeb database, a key resource on ant biodiversity, currently including more than 700,000 specimen records worldwide. From this, they were able to identify areas of high species richness and uniqueness across this broad selection of taxonomic groups, and thus to identify areas of the highest value for biodiversity conservation from among the candidate areas for protection. Then there was a coup d'etat, and this all kind of went out the window, at least for a while. But I think the lesson we should take from this is that as biodiversity scientists, we need to be ready when opportunity knocks to make data available to guide a decision-making process that can have profound positive or negative impacts on biodiversity. To better understand the taxonomic impediment and why it remains so difficult to practically apply taxonomic knowledge, especially for megadiverse taxa in regions with the most biodiversity, it is useful to discuss some of the factors that have inhibited the development of taxonomic knowledge. The Convention on Biological Diversity is an international agreement that came into effect in 1993 with 3 main goals: the conservation of biological diversity, the sustainable use of its components, and the fair and equitable sharing of benefits arising from genetic resources. While these are laudable goals, implementation issues have been disastrous for taxonomic research. A recent paper in the prestigious journal Science articulates the problem. The main issue seems to be regulations made in anticipation of financial benefits from biodiversity resources that do not differentiate between commercial research leading to proprietary rights and not-for-profit research such as inventories and taxonomic studies intended for the public domain. Biodiversity research has seemingly become suspect in the minds of many regulatory bodies, owing to fear that a taxonomic discovery today might conceivably translate into a commercial development tomorrow. Meanwhile, biodiversity is vanishing and scarce talent is walking away from research, so efforts to strengthen the third goal, equitable benefit sharing, have weakened the first goal, conservation and the development of biodiversity knowledge required for wise policymaking. I was recently in Thailand collecting specimens for a research project in my lab. As part of the process of applying for research permits, we were asked to provide a list of the species we were going to encounter and a count of the number of specimens we were going to collect. Such requirements are typical in many high biodiversity countries around the world. As I think you can probably understand by now, this is totally unrealistic, especially for research on megadiverse, poorly known taxonomic groups, and is incompatible with the whole idea of scientific discovery. In order to conduct research, we first have to report what we are going to discover. Meanwhile, land use changes, such as the conversion of forests into oil palm plantations, is proceeding rapidly. Industry does not need to report the number of species and their abundances lost when thousands of hectares of forest are cut down. It's only when we want to preserve specimens so that we can learn about biodiversity and share that information through publications and databases that we run into difficulties. Which brings me to another perennial issue in biodiversity science, the contribution of biodiversity research, specifically collecting specimens, to the risk of species extinction. Here is a recent example from a 2014 issue of Science that argues that scientific specimen collection is potentially harmful to many species, and that alternatives such as photographs and non-lethal tissue sampling for DNA are sufficient to document biodiversity. In fact, the most problematic contributors to contemporary species extinction are habitat destruction and impacts from invasive species. Molecular sequences and photographs, although useful, are not an adequate replacement for museum specimens, especially when it comes to newly discovered species. Not only is preserving specimens for science an extremely small source of mortality, We live in a society that kills billions of animals annually for food and by accident. Moral opposition to animal deaths can achieve much greater success with fewer downsides when aimed elsewhere. By comparison with other sources of animal mortality, scientific collecting poses a minuscule threat to wild populations in a time of global change when adding specimens to collections is now more important than ever, especially when it comes to megadiverse taxa for which taxonomic knowledge is far from complete. So I think a better way of looking at the moral dimension of specimen collecting is to promote an ethic that all specimens should be deposited in accessible collections with detailed data on the place and time of collecting, and to make that data widely available through publications and public specimen databases. I've talked quite a bit about the importance of names as the basis for organizing biodiversity knowledge. But there is an important caveat to that in this era of abundant DNA sequence data. Online archives of DNA sequence data like GenBank increasingly receive sequences submitted without scientific names. The interesting thing about DNA sequences is that they can be compared and grouped into clusters of similar sequences. Given the appropriate DNA region and analysis approach, these clusters can represent species-like entities. Whether or not these DNA-based species can be reconciled with the species of taxonomic nomenclature depends on whether at least some members of each cluster have identifiable voucher specimens. Copious DNA data accumulated over only the past several years have revolutionized our overall impressions of the biodiversity of some taxonomic groups. For example, an ambitious DNA barcoding study in Canada, based on 1 million insect specimens, found a number of species-like DNA clusters far in excess of the number of species known for Canada in some groups, like Diptera, the true flies, and Hymenoptera, the ants, bees, and wasps. A similar phenomenon has revolutionized our global estimate of the diversity of fungi. Traditionally, fungal taxonomy was largely dependent on the fruiting bodies, that is, the mushrooms, which in many species appear very irregularly. But the non-reproductive fungal tissue, the mycelium, are generally thought to be present even when the fruiting structures are not. It has recently become more common to assess fungal biodiversity based on samples of soil and other substrates using molecular techniques. The gap between 120,000 currently accepted species, according to formal taxonomic nomenclature, and estimates based on molecular sequencing exceeding 1 million means that fungi may be among the most underdescribed taxa, with only some 10% of species being known. We can think of the taxonomic impediment as a failure to get information from where it exists to where it is needed efficiently. You can think of this as the kind of supply and demand problem that economists might deal with. But if that's the case, how do we assess the value of information? And is it any different from the way we assess the value of physical goods? It turns out the development of a theory of the economics of information begins in the late 1960s in San Francisco with a band called the Grateful Dead. The band was known for its eclectic mix of styles, epic live improvisational episodes, and hordes of devoted fans who followed the Dead on tour. Among these fans were the Tapers, who recorded more than 90% of the Dead's live shows. Unusually for the day, recording Dead shows was not considered inappropriate. On the contrary, it was allowed, even facilitated by the band and their sound crew. The band encouraged exchange and distribution of these tapes, as long as this was done in a purely non-commercial manner. Some of the Dead's most enduring songs were written in collaboration between Dead co-founder Bob Weir and lyricist John Perry Barlow. The Grateful Dead were strongly associated with the culture of the political left in the U.S. in the late 1960s and beyond, a culture devoted to expanding civil liberties and questioning all kinds of social conventions. In the early 1990s, John Perry Barlow started to apply elements of this framework to the new world of cyberspace. He wrote a series of provocative articles for Wired magazine and other media, articulating why he thought that a free and unfettered internet was so important. It boiled down to his view of the economy of ideas and how the way we assign value to ideas differs from the way we assign value to physical things. His argument is encapsulated in the following passages from an article entitled "Selling Wine Without Bottles: The Economy of the Mind on the Global Net," which first appeared in Wired in 1993. He wrote, "In regard to my own soft product, rock and roll songs, there is no question that the band I write them for, the Grateful Dead, has increased its popularity enormously by giving them away. We have been letting people tape our concerts since the early 70s, but instead of reducing the demand for our product, we are now the largest concert draw in America, a fact which is at least in part attributable to the popularity generated by those tapes." With physical goods, there is a direct correlation between scarcity and value. Gold is more valuable than wheat, even though you can't eat it. While this is not always the case, the situation with information is usually precisely the reverse. Most soft goods increase in value as they become more common. Familiarity is an important asset in the world of information. It may often be the case that the best thing you can do to raise the demand for your product is to give it away. As scientists, ideas are our business and information is our product. So Barlow's economic paradigm may have something to teach us about how to maximize the value of what we do. Expressions of Barlow's paradigm can be found today in many corners of the internet. One of the most impactful has been the development of the Creative Commons license. These licenses allow others to copy and distribute the author's work with the stipulation that attribution to the original source is explicitly maintained. Optional clauses restrict commercial exploitation of the work, restrict modification of the original work, or require that derivative works be distributed only under the same Creative Commons license. So you could say that the counterculture that recorded dead shows has found contemporary mainstream expression in the Creative Commons license. The internet is great at making information much more universally accessible, so it has the potential to make primary taxonomic knowledge widely available. Keep in mind that academic authors operate under a different model than, say, book authors. Book authors are paid by publishers based on the anticipated or realized sales of books. Authors working in the academic world are generally paid by institutions which are themselves largely funded through public or philanthropic sources. So they typically receive no direct financial incentive to create content. Reviewers and often subject editors also participate in the publication process for no compensation from the publisher. It is just part of the professional obligation of an academic researcher. On the other hand, the public pays for a lot of this research indirectly through taxes allocated to public institutions and scientific grants. Of course, few things in life are quite so binary. Some important taxonomic research is conducted by people not employed by public research institutions. And of course, academics also publish books in the mode of many commercial publishers. But these are minority situations. Let's focus here on the primary mode by which taxonomic research is published, as peer-reviewed articles in scientific journals. In recent years, publishers of academic journals have taken two contrasting approaches. One model is to monetize research products. The alternative approach is open access publishing. Journals typically charge content creators a fee to defray costs associated with page layout, distribution, and other aspects of publishing. Most open access publishing is supported by fees to the author or their institution. With financial support levied in this way, articles become free for anyone online to read. The journals published by Pensoft, such as Zuki's, are particularly progressive and innovative expressions of open access publishing in taxonomy. The European Journal of Taxonomy, which is the product of a consortium of European natural history institutions, is another important open access platform for taxonomic publishing. In the practical exercise for this unit, you will be learning to use Golden Gate software to read and process taxonomic articles, such as those published in the journal Zootaxa, and share the content of those articles with Treatment Bank and ultimately the entire online world. So is Zootaxa an open access journal? No, it's a hybrid where authors have the option to choose whether to pay open access fees, thereby making the article freely available, or not, meaning that readers or institutions generally have to pay a fee for access. So some or all of you might soon be working with articles that are not open access. So is this practice even legal? Haven't I just roped you all into my sketchy cabal of taxonomy pirates? To better understand the legal interpretation of what Golden Gate and Treatment Bank do, it's useful to think about what copyright law is and what it applies to. It is true that most scientific literature is protected by copyright. Copyrighted works can only be copied, shared, and distributed when authorized by the copyright holder. This copyright protection typically expires some 50 to 70 years after the author's death. Copyright protects works of creative expression, especially in art and literature. The term work is key here. Although there is some variation in the way the term work is applied in different national legal codes, work broadly refers to an intellectual creation whose form of presentation is original, individual, and new. So copyright protection extends not to content, but to the form of presentation. Taxonomic treatments are principally collections of facts. Taxonomic treatment texts adhere to rules and predefined conventions. They are not works of creative expression in the sense of copyright law, and therefore belong to the public domain. The document created by the publisher is a form of presentation subject to copyright. What Golden Gate does is to extract those collections of facts, the treatments, from taxonomic papers. In this form, taxonomic treatments are in the public domain. They can be posted online, accessed freely, and shared with other compatible online resources. Up until now, we have emphasized the importance of specimen record data extracted from taxonomic treatments. But you might have already noticed that images, anatomical drawings, photographs, etc., that are cited in taxonomic treatments also appear on the relevant treatment bank page. Such images are conventionally treated as though they are the same as drawings or photographs created by an artist. But in fact, there is a big difference between a work of aesthetic and creative expression and the documentation of anatomical facts. Like the text of a taxonomic treatment, anatomical images in taxonomic papers follow predefined rules or conventions that facilitate comparisons with similar illustrations. They are not creative or original forms of expression. The more creative your ideas, the less useful they are for taxonomy. Taxonomists want to be able to compare images similar in view angle and technical style, even when produced by different authors, to facilitate comparison between similar species. This can be a difficult point to communicate to people because we are generally pretty used to the idea that creative is a good thing, and we all want to be thought of as creative. So if an image or piece of writing is not creative, that somehow seems to denigrate the object or its author. But seductive as this idea is, it is wrong, at least within the world of facts. Just to drive this point home, here we have anatomical drawings of skulls from 3 species of mouse, and also Mickey Mouse. The 3 sets of anatomical drawings repeat the same standard views and stylistic conventions to facilitate comparison between the species. Mickey Mouse is a work of creative expression created by Walt Disney and used to generate popular entertainment for decades. So Mickey Mouse is clearly protected by international copyright law, which reminds me, I better just blur that out a little bit. There we go. The anatomical illustrations of various mouse species are not creative and are useful resources for communicating facts about comparative anatomy. They are therefore in the public domain. Let's contrast the way Treatment Bank deals with the copyright issue with the way the Biodiversity Heritage Library does it. BHL, as it is commonly abbreviated, is the world's largest open-access digital library for biodiversity literature and archives. It provides free access to hundreds of thousands of volumes comprising over 55 million pages from the 12th through the 21st centuries. In addition to public domain content, BHL works with rights holders to obtain permissions to make in-copyright materials openly available under Creative Commons licenses. But here's the fine print. Most BHL content was published before 1923. This is because of a particular clause in U.S. copyright law that generally places content before this date safely within the public domain. More contemporary content is generally available only in exceptional circumstances, such as the receipt of explicit permission from the copyright holder. So what's the difference between BHL and Treatment Bank that causes these two resources to treat taxonomic literature so differently with respect to copyright law? The main factor is that BHL content is generally composed of scans of journal pages, typically organized as entire journal volumes. Treatment Bank does not distribute the original journal pages, only the facts extracted from those pages. And this brings us to an important development in the evolution of the World Wide Web. As Tim Berners-Lee will explain to you in a moment, the original idea of the World Wide Web was to be a place for all of us to put our documents so we could share them with each other. But the Internet is changing its focus from linked documents to linked data. This is sometimes referred to as the semantic web, and it is what you will be contributing to during the practical exercise with Golden Gate to mark up specimen records in the taxonomic literature. The XML tags you will apply contribute semantic enhancement to the original documents because they turn flat PDF documents into dynamic, exchangeable, reusable data. So here's Tim Berners-Lee, inventor of the World Wide Web, explaining the semantic web in 2009.
Time flies. It's actually almost 20 years ago when I wanted to reframe the way we use information, the way we work together. I invented the World Wide Web. Now, 20 years on, Ted, I want to ask your help in a new reframing. So going back to 1989, I wrote a memo suggesting a global hypertext system. Nobody really did anything with it very much. But 18 months later, this is how innovation happens. 18 months later, my boss said I could do it on the side as a sort of a play project. Kick the tires of a new computer we got. And so he gave me the time to code it up. So I basically roughed out what HTML should look like, hypertext protocol, HTTP, the idea of URLs, these names for things, which started with HTTP. I wrote the code and put it out there. Why did I do it? Well, it was basically frustration. I was frustrated. I was working as a software engineer in this huge, very exciting lab. Lots of people coming from all over the world. They brought all sorts of different computers with them. They had all sorts of different data formats, all sorts of kinds of documentation systems, so that in all that diversity, if I wanted to figure out how to build something out of a bit of this and a bit of this, everything I looked into, I had to connect to some new machine, I had to learn to run some new program, I would find the data, maybe the information I wanted in some new data format, and these were all incompatible. It was just very frustrating. The frustration was that there's all this unlocked potential. In fact, on all these disks, there were documents. So if you just imagine them all being part of some big virtual documentation system in the sky, then, say, on the internet, then life would be so much easier. Well, once you've had an idea like that, it kind of gets under your skin, and even if people don't read your memo, actually he did. It was found after he died, his copy, it was found he'd written vague but exciting in pencil on the corner. But in general, it was really difficult to explain what the web was like. It's difficult to explain to people now that it was difficult then. But then, okay, when TED started, there was no web. So things like click didn't have the same meaning. I could show somebody a piece of hypertext, a page which has got links, and we click on a link and bing, there'll be another hypertext page. Not impressive. You know, we've seen that. We've got things on hypertext on CD-ROMs. What was difficult was to get them to imagine. So imagine that link could have gone to virtually any document you could imagine. All right, that is the leap that it was very difficult for people to make. Well, some people did. So yeah, it was difficult to explain, but there was a grassroots movement. And that is what made it most fun. That was been the most exciting thing, not the technology, not the things people have done with it, but actually the community, the spirit of all these people getting together, sending the emails. That's what it was like then. Do you know what? It's funny, but right now it's kind of like that again. I asked everybody, more or less, to put their documents. I said, could you put your documents on this web thing? And you did. Thanks. It's been a blast, hasn't it? I mean, it's been quite interesting because we found out that the things that happen with the web really sort of blow us away. They're much more than we'd originally imagined when we put together the little initial website that we started off with. Now, I want you to put your data on the web. Turns out that there is still huge unlocked potential. There is still a huge frustration that people have because we haven't got data on the web as data. What do you mean, data? What's the difference? Documents, data? Well, documents you read, okay. More or less, you can read them, you can follow links from them, and that's it. Data, you can do all kinds of stuff with the computer. Who was here, or otherwise seen Hans Rosling's talk? When Hans Rosling's was at TED, yeah, one of the great, yes, a lot of people have seen it because it was one of the great TED talks. Hans put up this presentation in which he showed, for various different countries in various different colors, he showed income levels on one axis, and he showed infant mortality, and he showed this thing animated through time. So he'd taken this data and made a presentation which just shattered a lot of myths that people had about the economics in the developing world. He put up a slide a little bit like this. It had underground all the data. Okay, data is brown and boxy and boring, and that's how we think of it, isn't it, data? Because data you can't naturally use by itself. But in fact, data drives a huge amount of what happens in our lives. And it happens because somebody takes that data and does something with it. In this case, Hans had put the data together. He found it from all kinds of United Nations websites and things. He put it together, combined it into something more interesting than the original pieces. And then he'd put it into this software, which I think his son developed originally, and produced this wonderful presentation. And Hans made a point of saying, look, it's really important to have a lot of data. And I was happy to see that at the party last night that he was still saying very forcibly, it's really important to have a lot of data. So I want us now to think about not just 2 pieces of data being connected or 6 like he did, but I want to think about a world where everybody has put data on the web, and so virtually everything you can imagine is on the web. And I'm calling that linked data. The technology is linked data, and it's extremely simple. If you want to put something on the web, there are 3 rules. First thing is that those HTTP names, those things that start with HTTP:, we're using them not just for documents, now we're using them for things that the documents are about. We're using them for people. We're using them for places. We're using them for your products. We're using them for events. All kinds of conceptual things, they have names now that start with HTTP. Second rule, if I take one of these HTTP names and I look it up, I go and do the web thing with it, and I fetch the data using the HTTP protocol from the web, I will get back some data in a standard format, which is kind of useful data that somebody might like to know about that thing, about that event, who's at the event, whatever it is, about that person, where they were born, things like that. So the second rule is I get important information back. The third rule is that when I get back that information, it's not just got somebody's height and weight and when they were born, it's got relationships. Data is relationships. It's interesting, data is relationships. It's got this person was born in Berlin. Berlin is in Germany, and when it has relationships, whenever it expresses a relationship, then the other thing that it's related to is given one of those names that starts HTTP. So I can go ahead and look that thing up. So I look up a person. I can look up then the city where they were born. Then I can look up the region it's in, and the town it's in, and the population of it, and so on. So I can browse this stuff. So that's it, really. That is linked data. I wrote an article entitled Linked Data a couple of years ago, and soon after that, things started to happen. The idea of linked data is that we get lots and lots and lots of these boxes that Hans had, and we get lots and lots and lots of things sprouting. It's not just a whole lot of other plants. It's not just a root supplying a plant, but for each of those plants, whatever it is, a presentation, an analysis, somebody's looking for patterns in the data, they get to look at all the data and they get it connected together. And the really important thing about data is the more things you have to connect together, the more powerful it is. So, linked data. The meme went out there, and pretty soon Chris Bitzer at the Freie Universität in Berlin was one of the first people to put interesting things up. He noticed that Wikipedia, you know Wikipedia, the online encyclopedia with lots and lots of interesting documents in it? Well, in those documents, there are little squares, little boxes, and in those information boxes there's data. So he wrote a program to take the data, extract it from Wikipedia, and put it into a blob of linked data on the web, which he called DBpedia. DBpedia is represented by the blue blob in the middle of this slide. And if you actually go and look up Berlin, you'll find that there are other blobs of data which also have stuff about Berlin, and they're linked together. So if you pull the data from DBpedia about Berlin, you'll end up pulling up these other things as well. And the exciting thing is it's starting to grow. This is just the grassroots stuff again, okay? Now let's think about data for a bit. Data comes, in fact, in lots and lots of different forms. Think of the diversity of the web. It's a really important thing that the web allows you to put all kinds of data up there. So it is with data. I could talk about all kinds of data. We could talk about government data. Enterprise data is really important. There's scientific data, there's personal data, there's weather data, there's data about events, there's data about talks, and there's news, and there's all kinds of stuff. I'm just going to mention a few of them so that you get the idea of the diversity of it, so that you also see how much unlocked potential. Let's start with government data. Barack Obama said in a speech that American government data would be available on the internet in accessible formats. And I hope that they will put it up as linked data. That's important. Why is it important? Not just for transparency. Yeah, transparency of government is important, but that data, this is the data from all the government departments. Think about how much of that data is about how life is lived in America. It's actually useful. It's got value. I can use it in my company. I could use it as a kid to do my homework. So we're talking about making the world run better by making this data available. In fact, if you're responsible, if you know about some data in government department, often you find that these people, they're very tempted to keep it, to Hans calls it database hugging. You hug your database, you don't want to let it go until you've made a beautiful website for it. Well, I'd like to suggest that right before you, yes, make a beautiful website. Who am I to say don't make a beautiful website? Make a beautiful website, but first, give us the unadulterated data. We want the data. We want unadulterated data. Okay, we have to ask for raw data now. And I'm going to ask you to practice that, okay? Can you say raw? Raw. Can you say data? Can you say now? Right. Raw data now. Raw. Practice that. It's important because you have no idea the number of excuses people come up with to hang on to their data and not give it to you, even though you've paid for it as a taxpayer. And it's not just America, it's all over the world. And it's not just governments, of course, it's enterprises as well. So I'm just going to mention a few other sorts of data. Here we are at TED, and all the time we are very conscious of the huge challenges which the human society has right now. Curing cancer. Understanding the brain for Alzheimer's, understanding the economy to make it a little bit more stable, understanding how the world works. The people who are going to solve those are scientists. They have half-formed ideas in their head. They try to communicate those over the web, but a lot of the state of knowledge of the human race at the moment is on databases, often sitting in their computers and actually currently not shared. In fact, I'll just go into one area. If you're looking at Alzheimer's, for example, drug discovery, there is a whole lot of linked data which is just coming out because scientists in that field realize this is a great way of getting out of those silos because they had their genomics data in one database and in one building, and they had their protein data in another. Now they are sticking it onto linked data, and now they can ask a sort of question that you probably wouldn't ask, I wouldn't ask, they would. What proteins are involved in signal transduction and also related to pyramidal neurons? Well, you take that mouthful and if you put it into Google, of course there's no page on the web which has answered that question because nobody has asked that question before. You get 220,000 hits, no results you can use. You ask the linked data which they've now put together, 32 hits, each of which is a protein which has those properties, and you can look at the power of being able to ask those questions of a scientist. Questions which actually bridge across different disciplines is really a complete sea change. It's very, very important. Scientists are totally stymied at the moment. The power of the data that other scientists have collected is locked up, and we need to get it unlocked so we can tackle those huge problems. Now, if I go on like this, you'll think that all the data comes from huge institutions and it's nothing to do with you. But that's not true. In fact, data is about our lives. You log onto your social networking site, you pick your favorite one, you say, this is my friend, bing, relationship, data. You say, this photograph, oh, it depicts this person, bing, that's data. Data, data, data. Every time you do things in the social networking site is taking data and using it, repurposing it, and using it to make other people's lives more interesting on the site. But when you go to another linked data site and you say, this is one about travel, and you say, I want to send this photo to all the people in that group, you can't get over the walls. The Economist wrote an article about it. Lots of people have blogged about it. Tremendous frustration, the way to break down the silos, to get interoperability between social networking sites. We need to do that with linked data. One last type of data I'll talk about, maybe it's the most exciting. Came down here, I looked up on the OpenStreetMap. OpenStreetMap's a map, but it's also a wiki. Zoom in, and that square thing is the theater which we're in right now, the Terrace Theater. I didn't have a name on it, so I could go into edit mode, I could select the theater, I could add on down the bottom the name, and then I could save it back, and now if you go back to the openstreetmap.org and you find this place, you will find that the Terrace Theater has got a name. I did that. Me. I did that. It's the map. I just did that. I put that up on there. Hey, you know what? The street map is all about everybody doing their bit, and it creates an incredible resource because everybody else does theirs. And that is what linked data is all about. It's about people doing their bit to produce a little bit, and it all connecting. That's how linked data works. But you do your bit, everybody else does theirs. You may not have lots of data which you have yourself to put on there, but you know to demand it. And we've practiced that. So linked data is huge. I've only told you a very small number of things. There are data in every aspect of our lives, every aspect of work and pleasure, okay? And it's not just about the number of places where data comes. It's about connecting it together. And when you connect data together, you get power in a way that doesn't happen just with the web, with documents. You get this really huge power out of it. So we're at the stage now where we have to do this. The people who think it's a great idea, and all the people, and I think there are a lot of people at TED who do things, because even though there's not an immediate return on investment, because it'll only really pay off when everybody else has done it, they'll do it because they're the sort of person who just does things which would be good if everybody else did them. Okay? So it's called linked data. I want you to make it. I want you to demand it. And I think it's an idea worth spreading. Thanks.
The genius of Linnaean taxonomic nomenclature is to assign a unique identifier to every species on Earth. This is the mechanism that allows us to accumulate knowledge about biodiversity. Biodiversity is vast, one of the most information-rich fields of human knowledge. Advances in cyber taxonomy have only recently provided the tools to take on the taxonomic impediment and create a biodiversity knowledge network that is responsive to the questions of our time.